Hi, good morning, everybody. Welcome to the Polysystem Functions session. My name is Patricia Otera, and I'm going to be co-chairing this session with Dr. Anthony Maranto. We are both here at University of Maryland. Our first speaker is Dr. Cristina Cebrian. Dr. Cebrian is an assistant professor in the Division of Developmental Biology in the Department of Pediatrics at Cincinnati Children's Hospital Medical Center. Her lab has an established research program in kidney development, and her current goal is to understand the earliest cellular and molecular mechanisms driving autosomal dominant polycystic kidney disease. The title of her talk is Cisclonality and Identity in Autosomal Dominant Polycystic Kidney Disease. Cristina, you can take it away.
Thank you very much for inviting me to present our work here. I like to say that these kind of seminars are extremely important for those of us who are relatively new to the PKD field. And it's also very intimidating because I'm well aware that many of you have most probably forgotten more about PKD than I will ever know. But with that in mind, I am a developmental biologist by choice and by training. And my favorite organ is the kidney. And I hope I don't have to convince you that the kidney, it's a remarkably complex organ in which the structure of the organ and the function of the organ are intimately ligated. And how this structure develops or how this structure arises is what we are interested in my lab, in studying kidney development. So what you see here is what we are used to look at in the lab on a regular basis. What you have is mouse embryonic kidney explants that we've isolated at 12.5 days of development. And we can take the embryos out and put them in the air media interface and allow them to grow and visualize them. So we developmental biologists are very visual people, and we need to see how things happen. So what you have on the left is a reporter with HOXB7 that's labeling the entire collecting system. So the collecting system starts with this branching that happens at the tips of the ureteric buds. This is where the progenitors for the collecting that arise. And this branching is gonna keep happening throughout kidney development to generate the entire collecting system. On the right, what you can see is 6-2-Cre labeled with GFP in here. 6-2-Cre, it's a marker for the nephron progenitor cells. So these are the cells that are gonna give rise to the nephrons. And in here, you just see the progenitors that self-renew. They remain at the tips of these ureteric buds, and they self-renew to give rise to the entire nephron endowment in the kidney. Now, it is not that we developmental biologists are not exposed to cysts. We are quite familiar with them. There are several mouse models that develop cysts either at newborn or shortly after. In this case here, this is a TAS model in which this is from the McNeil lab that they've removed TAS in the nephron progenitors, and you see all these cysts developing. This work, I believe, is from Tom Carroll's lab, also removing Wnt9b from the entire animal or just from the collecting system generates cysts. And even our own data from our lab, we have a mouse model in which ETB4 and 5 are mutated from nephron progenitors, and we also observe cysts. Now, I'm a little bit embarrassed to say that in the developmental biology world, we look at this cyst and we say, well, defects in proliferation, defects in planar polarity, maybe a connection issue, and we don't go into much detail studying the cyst. And then I start learning about PKD, and I start talking to you. And again, I am amazed by the level of sophistication with which you look at this cyst. So it is overwhelming. It's hard to look at it because you have cell biology, you have everything about the cilia. Just the cilia on its own will give me a headache for years. But I had to dumb it down so I could understand this process in a very simplistic way. So this is how I see ADPKD for dummies. The first thing is that this is a disease that is dominantly inherited. Patients are heterozygous for mutations in PKD1 and PKD2. And I was very surprised to learn that there's a heated but very healthy discussion on whether there is a second mutation that gives rise to the cyst, or if it's just a decrease in polycystin activity. In both scenarios, what happens with this second event, I'm not even gonna call it a mutation, second hit, it's the second event, is that some cells fall below a threshold of polycystin activity, and those are the ones that are gonna become cystic. The other aspect of ADPKD that I found extremely interesting was the supposed clonality of the cyst, namely that all the cells in the cyst arise from a single mutant cell. And it was hard for me to find a lot of information about this detail, and I found this extremely interesting report that it's nearly 20 years old here. But in this experiment, and it resonated to me because they were developmental biologists, they were using morulus in here. They made blastocyst recombination. They combine ES cells that are PKD mutant with a morula from an animal that is PKD heterozygous. However, it is labeled with beta-gal. So what you see in here, every single cell that's blue, it's wild type for PKD1. Every single cell that is heterozygous for PKD1. Every single cell that does not have LAG-Z, it's a PKD1 homozygous mutant. So what they observed was that very early, the cyst had a lot of non-mutant cells. And then over time, the cyst will grow and all the cells will remain mutant and the wild type cells will be eliminated. They were even showing these hybrid cysts in here in which several wild type cells were in the same tubules as these mutant non-labeled cells, The way they interpreted these results is that one or a few mutant cells are capable of generating the cyst, and that then these wild type cells were eliminated and taken over by the mutant cells. While this was a very compelling report, the main issue with this approach is that the mutant cells were there from the very beginning. At the very beginning of kidney development, you had a lot of representation from those cells that don't necessarily recapitulate the idea of you get a second hit in one of these tubules. So the other aspect that I found amazing about ADPKD is the concept that the cysts are isolated, or maybe I should say they are self-contained. It is not a dilation of the epithelial tubule, it's a cyst that is a little ball that grows from the kidney. And I have to say, I didn't find many reports on how this actually happened. Do you get a large tube that then becomes a ball, or do you start with a very tiny cyst? The fact that nephron progenitor cells, the cyst could arise from all nephron segments, that's actually interesting, but there's very little information out there on whether all tubules are equally likely to become mutant upon a second hit. And also the fact that the cysts are asynchronous. So you don't get them all at the same size. They don't develop all at the same time. You have patients with smaller cysts and larger cysts. And that is not necessarily what we see in the mouse mutants. These awesome reports from the Germino lab, both in the rapid progressing, so when mutations are induced during development, but also in the slow progressing disease, you end up with a kidney that is all of it mostly cystic. So it's hard to see in the PKD1 and the PKD2 mutants, a progression of cyst growth. So as I said, as a visual developmental biologist, what I was really interested in was to understand whether the clonality was real or not, where the disease, how do they become isolated, and what's the representation of the different nephron segments in the cyst that is observed in the disease. So we thought about generating a new model that had to have, first of all, target a limited number of cells. We cannot make an animal that has all mutant cells because we get a severely cystic kidney. So it has to be a limited number of cells. But also we have, because it's a limited number of cells, we have to be able to follow those cells over time. So it needs to be lineage labeled. And finally, it would be ideal if we could recapitulate this asynchrony of the human disease, so we don't induce the cysts all at once, but they are induced in a staggered way. Now, the model that we've generated, it's a relatively complicated genetic model, and I'm gonna try to drive you through it. But even if you don't understand this slide, the end product of it, I hope it's gonna be much easier to understand. But basically, this model is based on two hybrid proteins that are on the two sister chromatids, in this case of chromosome 5, which is where PKD2 is located in the mouse genome. So what you have is the N terminal of GFP separated by a LOXP site from the C terminal of TD tomato. In the sister chromatid, you have the N terminal of TD tomato separated by a LOXP site from the C terminal of GFP. One of these alleles, one of these chromatids carried the mutant allele for PKD2. Now, in the presence of CRE, think about it. If you recombine this CRE, and this is interchromosome recombination, it's very rare. It's not going to happen in every single cell. It's only going to happen very rarely. But if that happens rarely, what is going to happen is that you're going to reconstitute the green, you're going to reconstitute the red. You're going to have a cell that is yellow, but it remains heterozygous for PKD2. Now, if recombination happens in the G2 phase, while the cell is divided, and then you have the CRE recombining, you're gonna generate this situation. And now the segregation of these chromatids can either happen as a Z segregation, in which 1 and 4 and 2 and 3, are inherited separately. And what you generate is a cell, a daughter cell that is not labeled, and a daughter cell that is both TD tomato and GFP positive, but heterozygous for PKD2. However, and this is where it gets interesting, if the segregation happens 1 with 3 and 2 with 4, what you generate is two daughter cells in which one of them is labeled with TD tomato and homozygous wild type for PKD2, while the other cell is GFP positive and homozygous mutant for PKD2. The important points in here is that if recombination happens again in these cells, either during mitosis or before mitosis, that is not going to change the genotype or the color of the cell. So once a cell becomes red, remains homozygous wild type and remains red. Once a cell becomes green, remains homozygous mutant for PKD2. And also, if that happens in here on the other, if that happens in the other scenario, you can still have a non-labeled cell that becomes mutant and a labeled cell that becomes a yellow cell that can become green and red. So again, it's a relatively complicated model, but the way it works, the end product is that in a heterozygous tube, you're gonna generate either cells, your recombination, these rare recombination events are gonna generate cells that are either unlabeled or yellow, and both of them will be heterozygous for PKD2, but they can also generate cells that are green and red. And in this case, they are PKD2 homozygous mutant or wild type. And also the litter mates of these animals that don't have PKD2 will also generate green and red cells, but in those cases, all those cells will be wild type for PKD2. All right, so with this model in hand, we can now start answering a few of those questions that I mentioned at the beginning. Are the cysts really clonal? And how does the process of isolation of the cyst happens? So we started with a developmental model with the 6-3 that, as I mentioned before, labels the cells in the nephron progenitor. So we will be generating cysts in the nephrons. And the first thing that surprised us was how little recombination we got in these animals. This is a control litter mate and a vibrotome section, 500 micron vibrotome section of a control litter mate, and we don't see that many recombination events. This is very useful to study clonality, though. An interesting aspect here, we see mutant cells in everything that's derived from the nephron progenitor cells. So here you have this very nice glomerulus in which several, a nice proportion of the podocytes are labeled. Now, in the mutant, when green means PKD2 mutations, we saw a reduced number of cysts per kidney. It's probably we see between 1 and 5 cysts in the entire kidney. The interesting observation is that when we image through those cysts, and we have consecutive sections so we can image through the entire cyst, we actually see that all the cells lining up the cyst are green. So at this stage, we can say that at this point in the cyst growth, these cysts are indeed clonal. However, and I know this one doesn't look like it, that's because of the higher magnification in the single plane. If we look at it, the entire cyst is lined up by GFP positive cells. But the other thing that we observe is that there are green clones here, some of them small, but some of them relatively large, that have not become cystic. So that actually makes us wonder whether all mutation events actually lead to a cyst. Now, the comments that we got from reviewers and collaborators was, this is nice, very nice model, but 5 cysts per kidney, this is gonna take you a while to get any quantification of what's happening in how these cysts develop. So we were in the search for a high frequency induction model. And that was a painful experience. We started with CMV-CRE, that gave us barely any recombination. The ROSA26-CRE RT2 inducible with tamoxifen, absolutely no recombination whatsoever. We tried specific CREs like KSP-CRE and Velen-CRE. Interestingly enough, the KSP-CRE gave no recombination at birth, but we got a lot of recombination after birth, and they rarely developed cysts. But finally, 2 models, the HPRT-CRE and the UBC-CRE, have given us a decent amount of recombination. So what you're looking at here is these 2 models, the control of the UBC-CRE, and you can see we get a significantly higher level of recombination. If we look at the mutant litter mates of those animals, and these are all 3-week-old animals, I hope I can convince you that there's, just in these 3 sections that I'm showing you, a significant number of cysts. Now, when we look closer at these cysts, what we can tell is that even the smaller ones, even the tiny cysts, are fully lined up by mutant cells. So in this Z-stack, you can see here, we have this interesting dilation in these mutant cells here and here. And this interesting also epithelial rounding up. So we believe, and we've confirmed that throughout several sections for both the UBC-CRE and the HPRT-CRE model, that even from the very beginning, the cysts seem to be made out of fully mutant cells. However, the ideal way to visualize this, and right now we are doing manual
2
annotation.
minutes.
Thank you. Would be to actually have a AI approach so they can go through these sections and visualize everything. However, we are still working on that, but sometimes we see events like this. So what we see in here, this is actually a glomerulus with podocytes and several cells in there. And in here, I was so glad when I saw this because I thought, look, we have so many tiny cysts. I hope you can see how many, oops, sorry. How many you can see them here without the movie. These little round things in here, but it turns out what we were observing in this animal was vessels, because this is the HPRT Cree actually animal, and we were labeling the blood vessels, and they gave this very nice round structure that had nothing to do with the cyst. So we are still working on the automatic annotation. As of now, we're just doing the manual annotation for these sections, for these sections. The other thing that we can do now is look at different markers within these sections to see what is the prevalence of the different tubules. So we are doing antibody and lectin staining for these specific segment markers. And again, we are staying with manual annotation until we develop the AI. But the consequence of what we can see is that we see a high prevalence of recombination in the collecting duct and the proximal tubule. So we see a lot of fluorescent cells in there, but we see a higher frequency of cysts in distal nephron segments. Again, since this is a manual annotation, I don't think our numbers are as solid as I would like them to be for now, but it seems like the distal nephron is more likely to develop a cyst compared to the mutant one. So with that, I'd like to thank everyone in the lab. This work was mainly done by Madison, but now we have Prashant and Kyla taking over this project. I wouldn't be able to do this without the help of the imaging core. There's many terabytes of data generating these images. And Simon Hippenmeyer, who generated the MADA model, and Kyle McCracken, which is a collaborator. We are trying to do an organoid model of this MADAM system. And with that, I'll be glad to take any questions.
Well, thank you very much, Dr. Cebrian. We do have a question in the chat. In the high frequency model, is there any cyst still connected to the renal tubule?
So in the high frequency model, only the very early ones, only the ones that we are labeling as dilations, slight dilations of the tubule, are the ones that are still connected. So they become isolated from the tubule very, very fast, very, very early during the cyst development.
Another question. Have you thought about injuring the mice to see if a third hit also causes clonal cyst formation or expansion?
We did that with the KSP Cree model. I don't know if I've mentioned that we didn't get cysts, we didn't get recombination at before birth. All the recombination we got, it was after birth, and they rarely developed cysts. However, if we injured them and we were using lithium chloride to actually induce a mild injury in the collecting duct cells, we actually observe the development of cysts. So yes, that's how the model works.
Do you see any phenotype in the blood vessels that are mutant?
No, we have not. We have not studied that in detail. And that's also something that I want to mention here. Now, with the HPRT and the UBC Cree, we are making mutant cells in every single cell in the animal. So we were considering using this to study other organs outside of the kidney, because we're generating mutant cells in there. We have not seen anything in the mutant vessels. They seem to develop normally, and these animals don't have any renal severe defects.
I do have a question. You mentioned you were already aging the mice for like 3 weeks. Have you already waited longer to see?
So for the 6 to 3, we've waited very long. The ones that have only very few cysts, we waited very long because we wanted to see whether those clusters that are mutant but are not cystic, whether they would eventually develop cysts. The problem is that quantifying that with such a reduced amount of recombination is difficult. But in the UBC-CRE and in the HPRT-CRE, we have not, I mean, we have those animals, we have not analyzed them yet. We're letting them age, but it will be interesting. I mean, some of them don't make it that far. Some of them are in worst shape because everything is highly mosaic in those animals. But yeah, we have those animals set up to see what happens after age. We assume they're going to become much severely mutant.
We have some more questions.
2 minutes.
Yeah, thank you. I'm wondering for the expression imaging, it is acquired 3D to capture the whole kidney. It would be very interesting to register, align this expression imaging with conventional medical imaging MRI, for example, to start.
Absolutely. Yes.
To explore like
imaging. Yeah, 3D imaging of the entire kidney is where we want to go. We have optimized protocols for clearing the entire kidney. This is way easier at 3 weeks than it is at 3 months or later. But also, we didn't have the imaging capabilities or the perfect imaging capabilities yet. Our institution has just acquired 2 light sheet microscopes, and one of them is specific for large samples. So we hope within the next couple of weeks, we're gonna actually be able to start imaging the entire kidney and correlating that with what that MRI looks like.
I'm gonna try to fit 2 more questions in the last minute. So Dr. Germino is asking, in the injury model, where you saw increases cysts, were they clonal mutant due to enhanced meiotic recombination?
They were clonal. I'm guessing that's the reason why they were clonal, yes.
Do you see glomerular cysts?
No, we don't.
Could DFP be secreted into the lumen and reabsorbed by nearby cells?
It could. I'm trying to think how frequent that will be, but I guess it could. I don't know how frequent that is. It will happen both for green and for red. So
yeah, and I think that's it. Thank you very much, Christina.
Thank
Very
you.
nice presentation. So Tony, you wanna go ahead with the next speaker?
Sure. Our next speaker is Dr. Luis Menezes. And Dr. Menezes is a staff scientist at the PKD section in the kidney diseases branch of the NIDDK. His work applies omics approaches to animal models with the goal of understanding the initial steps of cystogenesis. And the title of his talk is Omics Approaches to PKD.
Okay, thank you for inviting me. I'll share now. Can you see my screen? Okay. So I'm going to talk a little bit about omics approaches to PKD. And as you all know, transcriptomics is becoming an integral part of research. Here is just searching PubMed with transcriptomics. You see increasingly more people using it, up to 30,000 papers using it around 2020. And same thing for proteomics, metabolomics, epigenomics, came a little bit later. And we're now with the multi-omics, which allows you to combine all of these. In PKD, that has also been the case. So here I'm summarizing some of the studies that have been published. You can see the bars correspond to number of samples used, and you can just look at this summary here, see that some studies had large number of samples, some a little bit less so. Also, many models were used, so human organoid, mouse studies, rat studies, sex was variable. affected nephron segments were different depending on models used. Some were severe, some less so. Gene inactivation efficiency also varied a lot depending on 3 models that were used. And also multiple platforms were applied for this, from gene arrays to RNA-seq, and more recently, single-cell RNA-seq. Despite all this variability, a few common themes have emerged. So if you look at a word bubble of abstracts of these transcriptomics papers, see that genes involved in proliferation, apoptosis, cellular metabolisms, cytoskeleton matrix modeling, and immune response have all been identified. But yet the question is there an early PKD or PKD1, PKD2 specific fingerprint that can be identified and would be early and cell autonomous? So just looking at our old microarray data set, if you try to identify early changes in pre-cystic samples, there were very few differentially expressed genes. So we wondered, well, could you find a cell autonomous fingerprint? And we looked at some cell lines. Those are pair cell lines, the control and corresponding in vitro inactivated mutant. And you can see that for this one particular cell line here, you find few differences between mutant and control. Similarly, derived cell line shows more differences. But then if you look at the same cell line and a few passages later, differences are not as pronounced. And now instead, if you just compare different passages within the same genotype, you have many more differences here than you saw before in genotype. And of course, if you now compare same genotype but across these different cell lines, which are supposedly identically derived from similar animals in the same colony, same protocol, they actually have many more differences than just what we saw here in this condition. So what's the hope then of finding a PKD1 specific fingerprint within all this noise? So one alternative would be to look at meta-analysis, trying to see what's common between different models. And this too has a few challenges. So I'll go through an example using 3 recently published data sets. So I'll be using the ADPKD human sample data set published by the Humphreys lab that includes 5 cystic samples and 3 control samples. A PKD1 mouse model single-cell RNA-seq data set published by the lead lab at Mayo. It includes 3 time points from pre-cystic to cystic, 1 mutant and 1 control pools of 3 mice for each condition. And human PKD2 organoids, this is bulk RNA-seq done by the McMahon lab, and includes many samples. I'm just restricting the analysis here to 3 controls and mostly the 3 cystic mutants. So one of the challenges of here is just comparing identifying the cell types, and you can see that the cell type heterogeneity between these models can be high. So for example, you can see that the organoids here have, not surprisingly, lots of developing nephron cell types. In the human data set, you see lots of convoluted tubules, which you don't see as much in the other models. And in the mouse model, for example, you see connecting tube segments that you don't see as much in the human model. This heterogeneity also happens even within these groups. So for example, if you look here just at a human data set, looking at principal cells, you can see that majority of the cells are mutant, and within these mutant cells, large percentages coming from 1 sample only. Similar thing in the mouse. So again, just looking at the principal cells, you can see that in KOs, that most of the principal cells are coming from KOs, and again, again, mostly from one sample. And now, even if you restrict to just one cell type, so here is just focusing on leukocytes in one of these studies, and you can see that a lot of the clustering here is explained by sample of origin. So here, different clusters are colored differently, and they are coming from, they're colored by the sample of origin. So still one would hope that if you compare the differences in control, wild type in all these models, you'd find an overlap here that would be meaningful. But of course, the problem becomes then what percentage of this overlap would actually be meaningful, and it becomes even greater problem if you consider the rate of false positives with too many perhaps differentially expressed genes. So one alternative we thought about is that you could maybe add data from orthogonal data sets to try to interpret the differences that you find. And towards this goal, we had started making a PKD1 EGFP knock-in model that we could use for affinity purification mass spectrometry proteomics. And we use CRISPR to introduce EGFP3AJ tag after exon 46, which would result in PC1 with a EGFP3AJ tag in the C terminus. You can see here. So we made this model and we bred it to PKD1 KO heterozygous mice, and we saw that the animals were born at expected Mendelian rate. So you can see here the PKD1 KO and knock-in, and the corresponding PKD1 KO wild type, a similar number of pups. We also aged these animals, and they had no survival issues. And we looked at the kidneys, which had normal morphology, suggesting that PC1 EGFP 3AJ is functional. We also looked at the expression and saw that this was expressed. So this is P1 pups, mouse pups, and showing some of the organs that had higher expression. This is the whole head, lung, heart, kidney, and you're seeing the many PC1, so the full length, which is a whole protein, the CTF, which is this part here, and CDT, which includes lots of fragments, and another P100 band, which is we also use this for immunoprecipitation. We're using GFP nanobeads, which was quite effective in pulling down PC1, and you see these same bands here. So we then decided to move on and do AP mass spectrometry using the PKD1 knock-in mouse. And we tried this with kidneys, but eventually realized that the amount of protein that was needed for successfully detecting the bait was very high. So we resorted to using heads, which we could, they were bigger, they also had higher PC1 expression. So as an initial approach to this problem, we used heads of PKD1 wild type in EGFP animals. And we used 2 heads, then did IP of these 2 heads, combined 4 IP experiments like this, and then this collected sample set of 8 samples would be run as a 1 mass spec experiment, which we repeated 3 times. One for the PKD1 EGFP, EGFP as the sample that was IP'd, and also the same thing for a control group in which we IP'd PKD1 wild type for comparison. So we, on purpose, because of our preliminary pilot studies, decided to use kind of low stringent washes to enrich for the amount of proteins that we could pull down. And with that approach, we end up pulling down lots of proteins also in the control group. And we then look at the ratios of how much is pulled down in the EGFP compared to the control, an approach which had been used by other labs, including the Mann lab. And as you can see here, this was was successful in pulling down PC1, and the most abundant partner that we then detected was PC2, not surprisingly. What was a little bit more surprising is that we actually saw very few proteins that were enriched to the same level. So we had these two and then everything else. And this everything else sometimes was enriched in the EGFP, sometimes not, and these would labeled here in green, and I'm labeling here in purple the ones that were always enriched with a ratio above 2 in the EGFP condition. We can see here, we can see here that is actually pulled down a lot more. We enriched in the EGFP condition, not surprisingly, but also a few were in the control group. So to look at what was potentially part of the PC1 interactome, we again decided to follow something that had been used by the Mann lab. And that's basically, we looked at these ratios and suppose that everything that had a ratio of enrichment above 1 in the EGFP conditions compared to the control in all experiments was likely to be a true interactor, or directly or indirectly. And if it had a ratio of 2 in at least a few experiments, that would make it maybe a little bit stronger interactor. So we consider this set here, which is now labeled in green, as a potential PC1 interactor. And again, this could be interacting with PC1 either directly, indirectly, or transiently. And we then had a comparable set of proteins with similar kind of reverse criteria. So they were sometimes enriched in one or the other of the conditions and not very tightly bound. And this yielded about 991 proteins that we used as control. So if you look here, this would be the proteins in this PC1, putative PC1 interactome, and you see all the nodes. And if you now superimpose known protein-protein interactions that we pulled out from the string database, you can see that indeed they're highly connected, suggesting again that we are pulling perhaps a few PC1 direct binders plus their interactors in a network of proteins. So this interactome was comparatively enriched in plasma membrane proteins and also mitochondrial proteins. And looking at gene ontology, functional categories, we saw enrichment in metabolic functions, for example, oxidative reduction processes that were enriched in this interactome, but were not even represented in the background set of proteins. Likewise, if we look at pathways that could be enriched, we see enrichment in, for example, proteins related to glycolysis, DCA cycle, oxidative stress redox, PPAR signaling pathway, things that were previously allegedly involved in PKD. So we then looked at what we had started off to do, which is combine the transcriptomics with the interactome, and the hypothesis would be that perhaps you'd find some overlap that would be related to pathways that are regulated and modulate phenotype, regulated by PC1 modulated phenotype, and we saw this overlap here. And the question is this meaningful? And we hope it is, but of course it could also be by chance. If you just look at the overlap between the background proteins and the transcriptome, you also find reasonably high overlap. But it's possible that it's actually, this could be functionally relevant. And one indication is that if you look at these proteins and try to see what they are enriched in terms of pathways, you see that they have enrichment in many pathways that would make sense with PKD cystogenesis. But if you look at the corresponding set here, you find no functional enrichment at all. But then, of course, the question is, how do you validate? Because without validation, this is all a little bit meaningless. And there are different ways, again, of deciding what to do. So you could focus on like volcano plots, see what is most highly differentially expressed in all these models and choose some of these genes for validation. Or you could perhaps focus on genes, and this is genes that have multiple biological functions. So each dot is a gene here, and you can see that some are connected to multiple gene ontologies, so perhaps they are more central to cellular function. Or you could perhaps be interested in mitochondrial function or any other function depending on your initial hypothesis, and this would be, for example, genes that are related to mitochondrial matrix or mitochondrial or whatever. Or you could focus from the interactome part of things through to functional groups of proteins that are part of networks that seem to be meaningful. Or maybe you just choose a selection of these. But the point is that there are too many ways to choose for this to become practical. So one would need an in vitro platform for validation. And we started looking into this, trying to develop a method that we could use in the lab. And we decided to go for the organoid system. So we are now developing a PKD1 from progenitor cell and NPC-derived organoids. And what we would hope to find in an in vitro system for this sort of analysis, that it would mimic disease process. So here showing that it forms cysts in the mutant state, tubes in the control state. It would have low variability. So we developed these NPCs from many mice, and here showing a subset. All the KOs develop cysts spontaneously, all the wild types do not. Should be fast. So here we get a PKD1 conditional. We induce in vitro. After 9 days, we have cysts. If we don't induce, then they remain normal. And they should also be easy to manipulate, so we can overexpress and knock out things that will. So basically, the blueprint for what we plan to do next is use these in vitro models for omics plus perturbations, then correlate with the perturbations with the phenotypes. And for this, we should kind of have accepted morphological criteria, for example, is this cystic or tube? And then from a subset of what we found here, go to animal models, which should also be standardized across the PKD community, and look at time points that are widely used, and evaluate the phenotype also based on predetermined sample sizes and effect sizes. So the conclusion then is that transcriptomics meta-analysis requires few assumptions. So the PKD has a detectable transcriptional fingerprint. That is, fingerprint is shared by different models and cell types. This fingerprint includes relevant changes that cause the phenotypes. But it's also useful that we could have orthogonal data sets to add independent findings to help interpret these. This could be perhaps different cystic disease models or different organ systems or complementary information like proteomics, as I showed here, or phosphoproteomics or spatial transcriptomics. But ultimately, the value of any of these high throughput experiments is only real after validation. And for that, we can have multiple strategies to generate testable hypotheses, but we should end up in a consensus set of models and phenotypes for evaluation, using also a consensus set of validation criteria. And with that, we hope that omics data with robust validation can be then used to identify PKD mechanisms and therapy.
Time.
Yep, and this is the people behind all the work, and I'd like to thank the labs that generated most of the data that I presented and either deposited them or shared them. Humphreys Lab, McMahon Lab, Dane's Lab, and Germino Lab. And that's it. Thank you. I'll stop sharing now.
Okay, we have a few questions here. First one is, how many extracellular proteins were enriched by the GFP?
So I don't know the exact number, so I have to go back and look, but they were mostly not extracellular. So the way, they were mostly, as I showed, they were mostly in cytoplasmic actually, and then also others. But what was actually enriched compared to so if you look at what was pulled down in general, that was kind of overall distributed equally. What was more enriched were those two categories, and it was mitochondrial proteins and and plasma membrane proteins.
In that case, I'd like to tack on a question to that one. Among those, I mean, you said that there weren't many, but among those that were enriched, that were extracellular, can you mention any of the identities of them?
Yeah, so I don't know off the top of my head, but it's so that's all published. We have a very extensive set of tables in the supplements. So everything that we pull down is there. The other thing I would mention is that so what I showed is a putative interactome, and that's based on some selection criteria, which we think are meaningful, have been used before, but they're not necessarily the only way to look at it. So we also deposited the whole kind of the raw data and the data set without filtering out. So you could, depending on how you change these filters, you see more or less extracellular proteins, and you may look for your specific target. But again, this becomes more relevant when you have a way to validate. Otherwise, it's mostly speculation as to why they were there.
Next question is, what was the molar ratio of PC1 to PC2 in the IP?
So I don't know. So for the mass spectrometry, we didn't have we didn't include any standards to calculate molar ratio. So what we saw is basically the ratio of the signals, and these signals are quite variable from experiment to experiment. So you normalize them internally. But in terms of the ratio of just how much it was enriched, then it was enriched from 10 to 30 times compared to the control for PC1, and about the same for PC1. So if you just compare the ratios, then they would be somewhat similar. But again, sort of the IP would, so you would perhaps be more efficiently detecting PC1 than PC2 in this condition.
2 minutes.
And could you determine the molar ratio of PC1 CTF to NTF?
So, well, again, so we would only pull down NTF that was bound to PC1. So the quick answer is no, I could not. So what we did is we looked to see if there was enrichment of, for example, are we pulling down just the C terminus and not the full length, because that was a concern. So what we did is we mapped all the peptides that we pulled down across the PC1, and we saw fragments from every fragment, but we saw more fragments coming to the more peptides from the C terminus. Whether that's because we are also IPing a little bit more of that after it's already cleaved, versus it's just because of how the peptides fly, I'm not completely sure.
It's another question. So given the high prevalence of many CTT fragments, do you think that the database may be enriched with CTT interactions?
Yeah, so that's what I was saying. So it's possible that it is, but it's also, I mean, we also pulled the full length.
There's a comment, 823 hits seems like a lot. Could you play with the stringency of your co-IP conditions to enrich for a stronger candidate?
We could, and I guess this is what one could do it, and that's why we have the data set open with, so people can play with all these things. But the point that we wanted to make is not so much that we would find. So we're hoping that perhaps we would find something like PC2, and up there some other target, and that would be a main binding target, which we didn't. So we then see this haze of things that are pulled down, and we want to make some sense out of it. So the approach we took is we look at this from a network perspective. So even if there's some noise in there, at least we are likely pulling down things that have interactors and would have would be functionally meaningful.
Okay, thank you. There are a few more questions in the chat, and if you want to address them on the chat, sure, they would be appreciated.
Thank you. We'll do that.
Okay, next, thank you very much to Dr. Meneses and Dr. Cebrian. Next, we're going to have like a short presentation from Dr. Muthasamy Tangarayu from Medical College of Georgia. And his talk has the title Netrin-1 Deficiency Limits Cystogenesis in Autosomal Dominant PKD. Dr. Zaffarani?
Yeah.
Yeah, okay.
So how do I share?
In the bottom, there is a green button that says share screen with an
Yes.
arrow. Just click there and you should be able to share your screen.
Okay, so the desktop. I have it in the desktop. It says allow Zoom to share your screen. Open system preferences. So you could not see my desktop. Okay.
Yeah, in the bottom, there is a share green button that say, if you click there, you should be able to share. Might probably ask you for permission and just click yes. And you should be able to share your screen.
Okay, files. Share. Open. It doesn't allow me to share the file. I don't know why.
So do you have your PowerPoint open already?
Yes.
It's open and minimized. So then you click the green share screen button at the bottom of your Zoom screen.
Yeah, that's what I did.
And it'll list a bunch of whatever applications you have open. It'll show them all. You just choose the PowerPoint.
Okay. Can you see?
Yes, perfect.
Okay.
Awesome.
Okay.
Okay, go ahead.
Hi, thank you so much for giving me an opportunity to present our work in the conference. I am new to the field because my lab is working mainly in the cancer research, mainly in the breast cancer and the colon cancer. And we started this work maybe in 5 years before, when we collaborated with Dr. Xia Hong Li in Mayo Clinic and Dr. Peter Harris and Dr. Thomas Wimes in the University of California, San Francisco. So we are working. We are working in 3 or 4 different project in ADPKD, which is also we are working in the cancer biology. The first one is in the role of GPR109A, which is a receptor for the niacin and the butyrate and the beta hydroxybutyrate. The clinical one of the clinical manifestation of ADPKD is increased cyclic AMP and the reduced the calcium signaling. So the GPR109A play a critical role in the regulation of cyclic AMP and in the increased intracellular calcium. And also we are working on the role of epigenetic in the regulation of ADPKD cystogenesis. And in collaboration with Dr. Thomas Wimes at the University of California, and we are working on the role of STAT3 signaling in ADPKD. And the today's talk, we are working on the role of Netrin-1 in ADPKD growth and the and its functional significance in the therapeutic intervention. So the Netrin-1 is the lamin-related secreted protein. It is has 5 different receptors. The Netrin-1 binds with the DCC, that is deleted in colon cancer, and also 5A, 5B, 5C, and 5D. And the interesting thing is, if there is no netrin, the netrin receptor function as a pro-apoptotic gene, thereby it reduces the cell proliferation and function as the tumor suppressor. But once the Netrin-1 binds the receptor, it activates the receptor. That the receptor activation, what it does is it blocks the proliferatory and the growth promoting signaling and function as the tumor promoter. So Netrin is function as a tumor promoter, although it is discovered as axonal guidance, since which is involved mainly in the neuronal development. But in most of the neural development or the axonal guiding molecule is suppressed in the tumor cells or the tumor tissue. But the Nisshin1 is overexpressed in most of the cancer tissue, mainly because of it binds the receptor and once the receptor activation blocks the p53, and that leads to cell proliferation and the tumor growth. So this is the cartoon on the
2 minutes.
Yeah, maybe I can. This also show that in the Nisshin1 antibody reduce the signaling, reduce of tumor progression. So we took the normal and the apical tissue, which we got it from Dr. Darren Wallace, and we checked the NGN1 expression in the human tissue, especially in the apical tissue, the expression goes up. And also we checked the different receptor, the UNC5H2 expression also goes down in both mouse and the human apical tissue. And we use the biological sample from urine and the serum, and we check the NGN1 expression. It is specifically the NGN1 expression goes up in the apical patient serum and the urine. And to check what is the role of the NGN1, we developed, we obtained the NGN1 knockout mice from Dr. Patrick Maglin from the France, and the Cancer Research Center. And if we knock down the NGN1, it is completely rescue the apical cystogenesis. And also we use the humanized mouse monoclonal antibody against the NGN1. What it does when the antibody blocks the binding of NGN1 into the UNC5B specifically. So if we use that, then the NGN1 humanized monoclonal antibody, it is completely rescue the cystogenesis of the ADPKD. So we have done several work, but because of the very short talk, I put only the important finding of our study. So the conclusion of our study, increased expression of Nisshin1 in apical biological fluids can be used as a novel biomarker to monitor disease progression and the treatment response. And also NGN1 humanized mouse monoclonal antibody can be used as a therapeutic strategy, either alone or in combination with the therapeutic drugs like Salvaptan for the treatment of apical. I stop here, and I can ready to take any questions.
Okay,
Yeah,
oh, you want to say something?
Yeah, I would like to thank all my lab members and all the collaborators like Dr. Peter Harris and Thomas Wimes and the Darren Wallace, and also Patrick Maglin. She gave the knockout mice and the monoclonal antibody, and also thank the PKD RRC. He gave the pilot and the feasibility award. So I stop here and take any questions.
Thank you so much, Dr. Takaharo. If I don't see any questions in the chat yet, but I do have a question. Okay. So how do you think Nisshin1 is acting in PKD?
Since it is a secreted molecule, it is in the proximal tubule cells. It expresses the Nisshin1. Once it is secretion, it is because of it's a secreted molecule. It is activate maybe the receptor, thereby to block the apoptotic signaling, especially the p53 and the other apoptotic molecule, and thereby might involve in the cell proliferation and the cyst development. Especially it binds with the UNC5B. So that is why in the antibody it is block the binding of Nisshin1 and the UNC5B, that block the cell proliferation. Maybe it activates p53 signaling. Maybe I'm not sure what is the role of p53 in the ADPKD, but in the tumor genesis, the p53 function as a tumor suppressor. But we have to look for what is the role of p53 in the ADPKD.
Mm-hmm. So I do have a question. So you mentioned that the expression of Netrin-1 was was highly expressed in urine and plasma of PKD patients. I was wondering if you were able to differentiate between males and females, and actually if you were actually able also to classify them based on the progression of the disease. Do you see the same high level of expression early on in the disease, and as well as later on, or it's already variable?
Yeah, actually, we did in the mouse, we could not find the significant difference in the male and female, but in the human, we got very few samples from Dr. Wallace. So we have to get more samples to differentiate whether is there any gender-specific variation in the Netrin-1, but in the mouse, we could not find. But in the transgenic mice, which specifically overexpress the Netrin-1 in the kidney, that the male mice only develop the disease, but not the female. Once if you do the overectomy or give the tamoxifen, you develop the cyst. But that is the kidney-specific transgenic expression develop the cyst only in the male mice, not in the female. But we could not see that difference in the normal mouse models of ADPKD, whether we can see the increased expression of Netrin-1 in both male and female.
Okay. There is one more question. Is Netrin-1 in the exosomes?
We are not sure whether we have to. It should be, since this is a secreted molecule. It should be in the exosome also.
And have you checked feeding behavior in the mutants?
2 minutes.
NTN has a role in adipose and food intake that can impact cyst formation dramatically.
Since that is the NTN1 knockout mice, it is significantly block the cystogenesis. But we are not, since that is a flocked mice, since the global NTN1 knockout is embryonic lethal, we developed the flocked mice, and the kidney-specific knockdown significantly reduced the cystogenesis of the in the mouse models. We developed 3 different mouse models. In all of them, we do show the significant effect, both the NTN1 knockout mice and also monoclonal antibody against NTN1.
Okay.
I have a question. Would it be possible to check if the antibodies to the Netrin might be also causing resorption of the cysts, let's say by doing serial MRIs?
Yeah, maybe. Could be. Since we have just started the project and we have a lot of antibodies, since Dr. Patrick Maglin, he gave a very generous amount of antibody. Since they used that antibody in the clinical phase 3 clinical trial in triple negative breast cancer, and we are started the project, and we will work very soon to check for whether the antibody can cross the cyst, and whether we will collect the fluid and then we will check.
Okay. Thank you very much. Tony, you want to introduce Dr. Chang?
Sure. Dr. Fang Chang will speak on behalf of the molecular subcommittee. And as you know, he's a professor at the University of Maryland Medical School.
Okay. I'm trying to get the sharing. Which button should I get? Oh, I got it. Sorry. All right, let's get this. Okay, could you see and hear?
Yeah, perfect.
Good. Okay, I'm going to talk about what we do at the molecular subcommittee. So the mission of the molecular subcommittee is really twofold. One is to provide validated PKD1 reagents, that is the PC1, PC2, and FPC fibrocystin. And the second mission is going to develop new resources. Now, we are actually working on 2 fronts. One is has anything to do with antibodies, and the second branch is really to working on DNA vectors, expression vectors, or CRISPR vectors. Okay, now first, to show you what we provide. So these are the reagents which we have. You can find them actually in our website. So again, antibody, you can find, this is just a brief summary. You can find them in detail. And we provide antibody against PC1, and these are the antibody epitope you can find. You can find the description there, and PKD1, PKD2, and fibrocystin. And you can find what they are good for. But generally, our reagents is good for immunoprecipitation, Western blot, and so on. And they are not as robust as we hope for if you want to detect endogenous PC1 and tissue in situ. So therefore, okay, vector as well, you can find them, many of them. So therefore, we also are developing new high quality PKD1, PC1, PC2 antibody, nanobodies now to detect endogenous polycystins, proteins in tissue and cell. How are we going to do that? Are we going to do anything differently? Yes, we try to do things differently. Number one, we're going to generate rabbit monoclonal antibodies. And secondly, we try to do is to generate VHH nanobodies by phage play technology. We also use a different kind of antigen as opposed to previous approaches. We were using folded human PC1, PC2 fragments that were expressed in and purified from egg cell, mammalian cell. Now I have to say these products were provided by Erhu University of Utah. So the antigen we use are either a N-terminal fragment of PC1 or the entire or a fragment, larger fragment from an extracellular region of polycystin 1. For polycystin 2, we were using basically the PC2 tetramer embedded in lipid nanodisk. Okay, I'm going to show you briefly what we have done, and we're hoping that they are good stuff to be provided to you if you want to use them.
2 minutes.
Okay, so developing rabbit polyclonal anti-human PC1 ECR, extracellular domain antibodies. Okay, so this is a map of polycystin 1 cleavage GPS, and this is NTF. So the fragment I'm going to be showing you are antibodies produced by using this ECR fragment. Okay, this is the entire extracellular region. So I'm showing you here, we actually, in the course of the development, we have actually purified serum from 3 rabbits, okay, immunized with this fragment. And they were positive on Western blot. And now this is the NTF. Now, this is the recombinant protein, just for us to show whether this antibody is good or not. And they're also positive to other fragments. And we also show that they are active. I mean, one of them, all 3 of them, actually were able to detect polycystin in primary cilium if you express them recombinantly. Okay, this is MDCK cell. I'm sorry, IMCD cell. They are positive. We move forward with developing monoclonal anti-human PCR. I'm sorry, antibody. Again, using this same antigen, we were able to collect a bunch of them of the monoclonal antibody. They're directed to different region of NTF. And they are, this is just show you they are positive on Western blots, and this is just a few clone from this area. Yeah, they're positive, and they also are working great for on immunoprecipitation. Yeah, so this is the positive. So they are good, and we are testing whether they are good for immunofluorescence, and if they are, then we should have some promising candidate here. Okay. And then
Time.
Okay, nanobody, basically also promising, positive. I'm going to skip with it. And okay, and the second, briefly, we also developing new PCR expression constructs because they're big. You cannot simply manipulate them easily. So we have developed actually a novel new PC1 human cDNA introducing tons of silent enzymes that you can actually use to manipulate. This is for human. We have also for mouse as well. In the final slides, we also have developed new CRISPR-Cas9 vectors, either alone, human mouse, or in combination with IFT88, that is in the mouse. I'm going to stop here and taking any questions if you have. Oh, are you there?
Yes. So the idea here is to hear from the community, whether they need already additional resources, antibodies and vectors from the core, and also if you have specific questions about the resources that are already available.
Could you enable my sharing? Could you do that from your side? I'm sorry, I can't do it from here. Okay, great.
Yeah.
There's a
Perfect.
question in the chat.
Fan, do you have a timeline on the availability of the nanobodies?
For the nanobody, we have actually at the medium stage, we can produce them, isolate them, and we can do the binding, but we haven't yet produced in E. coli and we haven't tested really on immunofluorescence. So in terms of maybe a year, I'm not sure, because we have so many candidates. We now focus on the monoclonal rabbit, but nanobody was on the table.
Hi, Fan, thank you so much. How many monoclonal rabbit do you have?
We have 17, 18, maybe 20. We have mapped very too narrow area in the NTF. So basically, that could be further validated. For example, thinking about the organ culture. For yesterday's talk, like Zhong Wei Li, or even today's talk of Dr. Escher, that could be used for validation, for the validation on really native protein. Now, this is something we haven't done. And for Marcus, you could use it maybe to test whether this has an effect on your cilia channel activity.
You probably have already answered this, but for immunofluorescence detection with antibody, did you say that it was endogenous or IMCD overexpressed?
This is overexpressed. Recombinant polycystin 1, polycystin 2 in the cilium.
What is the status for the PKD2 antibodies?
PKD2 antibody, we have currently 3 monoclonal mouse antibody directed to the nanodisk, and they working very good. Western blot, we haven't tested IP, but they working on cilium on IMCD3 cell when recombinant protein is overexpressed. Seems to be also positive.
Yeah, one comment is that it will be very helpful to have antibodies that cover multiple regions to say cleavage events.
For PKD2, I couldn't see it.
Yeah,
For PKD2?
for PKD2 and PKD1. I guess for PKD1, you already have.
Oh, yeah, for PKD1 is easy. So, I mean, this time we focus on NTF and extracellular domain. We have also an antibody previously made, if you look at the database, is against CTF, probably at the top domain, which is extracellular as well. And they seem to working on immunofluorescence, maybe a little bit more difficult, but it may be working as well. So by using these two sets, you should be able to see the whole set, whole region of protein. I'm talking about NTF and CTF. Regarding PKD2 is a little bit more, we haven't done too much. So we even don't know to what epitope our monoclonal PKD2, the new one, is actually directing to. But the cleavage of PKD2 is less studied. So we haven't focused on that yet, maybe for the future.
Another question is the EA antibody also working for immunofluorescence?
Yes. However, it requires a special condition. It's not as good as NTF. All these antibodies, you can actually see that in live cell. This is the beautiful thing about it, the monoclonal antibody. Yeah. For EA, we need certain detergent, and we're actually optimizing it. We haven't done that before, but now we thought it actually could be made to work as well. And the good thing about this EA is that it is cross-reactive to human and mouse. Again, we testing really only on recombinant protein so far. Yeah. Now, whether you can see endogenous. Now, if you say, "Oh, I cannot see it," now that could be two reasons. Either our antibody is not potent enough. Okay, so, or our condition is not optimized so that the epitope is hidden. So we need to do optimization. Tissue to tissue is different. Another reason would be is really that antigen is really so low. No matter how hard you try, you cannot get it. And we hope that using a panel of the new antibody, maybe one of them is potent enough that allow us to really see them above the threshold. That's the hope. And so same thing for an antibody.
If there are no more questions, we can move on to the lunch.
Yeah, so we're gonna give a full half hour. Let's just meet after lunch at 1:15. We'll start, okay? And thank you, everybody. Thank you to all the presenters, all the moderators. The great morning. Thank you.